Hello students, welcome to my biology class. This session is for students of class 12. Our discussion today is on chapter 5, Principles of Inheritance and Variation. This is the second topic which will be dealt in this chapter. Before this we have discussed Mendelism, and now I will be discussing some deviations from Mendelian monohybrid ratios. So welcome to this session where we will be discussing some deviations of Mendelism. In our previous discussion, we have seen Mendelism and the ratios, monohybrid ratios, 3:1, which is the phenotypic ratio, and 1:2:1, that is the genotypic ratios. Now, what are we going to learn here? At the end of the session, you will be able to discuss the causes of incomplete dominance, write the results of codominance, relate one example of codominance with multiple allelism, apply the concept of multiple allelism in ABO blood grouping. You will be able to solve some problems asked on blood group inheritance. Now, when experiments on peas were repeated, means Mendel was doing the experiments using different traits, he found certain observations which he recorded. And on the basis of the observation, he formulated the laws. But when some other persons were doing the same experiments, they found certain deviation in the F1 also. What they found? They found that the F1 generation, which Mendel had suggested, should be same as or should be similar to either of the parents. It was not found. Rather, they found some different phenotypes in the F1. In this session, we will discuss certain examples which deviations they had found. Let's start with incomplete dominance found in snapdragon. This is a common garden plant, Andrenum species. Here you can see these are the flowers. A very common winter flower. Here a red color flower is crossed with a white color flower. The observation is that in the F1 the flowers are neither red nor white. Rather, it became pink, which is a deviation from Mendelian concept. When these pink flowers are crossed, then what happens? Then in the F2, red simultaneously, pink flowers and the white flowers, they were obtained. Now let us try to understand. So Andrenum can be considered as a good example to study incomplete dominance. Why incomplete? Because here dominance is not complete. That is why the red color or the white color totally did not mask the other colors. Now here what we are to study, that is, he had taken, or the person who had made the cross had taken true breeding red flower and true breeding white flower plants. Let us denote them as capital R, small r, smaller. Now here you may ask a question that here am I writing that the white flower is recessive trait because I am writing smaller? It's not like that. This is true. This type of notations we write in Mendelism, where capital R is a dominant trait and small r is a recessive trait. This is just to make this thing clear we are writing. Now when such flowers are crossed, the F1 of course will be capital R and small r, and this F1 is neither red nor white, rather it is an intermediate, that is pink color. Now when the F1 plants are self-pollinated to get F2, in Mendelism the F2 phenotypic result was 3:1 and the genotypic result was 1:2:1. Here a deviation has been found. What is the deviation? The F2 phenotypes are 1 red, 2 pink and 1 white. So this is a deviation from Mendelism. Here, does the law of segregation apply? Yes, law of segregation applies. How? Because if capital R and small r does not segregate, then from the pink flower you can't expect red flowers and white flowers in the F2 generation. Since capital R and small r they separate during gamete formation, so new combinations of capital R and small r are possible. So red flowers and white flowers are observed or obtained in the F2 generation. That is why law of segregation here applies. Now, to give an explanation, here capital R is not completely dominant over small r. That means red is not completely dominant over white. And that is why the pink color is obtained. Since in a diploid organism, there are two copies of every gene, that is, the alleles are in pairs, these two alleles need not be identical. It can be homozygous, it can be heterozygous. If it is heterozygous, it will be capital R small r, for example. Now, let us take an example of a gene. Now, actually, what does the gene do? Yes, the gene expresses a character. Very good. Now, how does the gene express a character? The gene can prepare only or synthesize only a polypeptide. And that polypeptide may be an enzyme also. Let us consider that here the gene contains the information to produce an enzyme. Now, since there are two copies of this gene, that is, there are two allelic forms, let us assume that the normal allele produces normal enzyme. And this normal enzyme is required for transformation of a substrate. Let the substrate be designated as S. So, the normal enzyme produced from the normal allele will be catalyzing the transformation of a substrate S. Now, theoretically, any modified allele could be responsible for either of the three. We have already talked of normal allele. Now let us think of a modified allele. How does a modified allele work? It can be responsible for production of either the normal enzyme, or it can produce a less efficient enzyme, or it can produce a non-functional enzyme, or it may not produce that enzyme at all. So these are the different possibilities. Now in the first case, the modified allele is equivalent to unmodified allele. That means they have the same phenotype. If it is so, then the result will be that there will be a transformation of the substrate S. That means here, since the modified allele and the unmodified allele, they produce the same phenotype. But if the modified allele produces a non-functional enzyme or no enzyme, then what will happen? Naturally, the phenotype will be affected because there will be no transformation of the substrate. The phenotype of the trait will be dependent on the functioning of the unmodified allele. Here this is very important. The unmodified allele, which represents the original phenotype, as we have discussed earlier, the original phenotype here is the dominant allele, and the modified allele is the recessive allele. Hence, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. This is how we can explain such events of how the alleles are functioning. Now let us go to another deviation, that is codominance. We have discussed incomplete dominance. Red and white produces neither red nor white in the F1, rather it produces a different color, that is pink, which is intermediate of the two. Now let us study codominance. Is there a difference between codominance and incomplete dominance? Obviously, there is difference. We'll see here. Now, what happens in codominance? In codominance, the F1 generation resembles both the parents. That means both the alleles are expressing in the F1. That is why the F1 can resemble both the parents. One very good example is the ABO blood grouping in human beings. In ABO blood grouping, only the AB blood group shows codominance. Why? Because in such AB blood group people, they have antigens, both A antigen and B antigen. We'll discuss more later on in multiple allelism. But here, let us discuss at least that in human, there are blood groups A, B, AB and O. In AB, both the antigens are present. That is, they have alleles for antigen A, have antigens for allele B. Here, both the allele A and allele B are functional. That is why it is an AB blood group. In O blood group, no antigens are produced. Whereas in A blood group, antigen A is produced, and in B, antigen B is produced by the allele. There are three alleles, IA, IB, and small I. The combination of these three makes the blood groupings. We'll discuss more in our different topic on multiple allelism. Now, here you can see a beautiful picture. This is Andalusian fowl. Can you see here? This is black color, white color, and a blue color. Codominance is very well studied in Andalusian fowl. Let us study codominance in Andalusian fowl. There are three variants of this fowl. One is splashed white, another is black, and the third is blue. When a pure splashed white male and a pure black female is crossed, they produce an hybrid, and the hybrid is blue color. So the F1 is different from the parents. F1 is blue and the parents are white or black. Now the question arises, how this blue color comes? Is it due to mixing of the colors? No. Here there is no mixing of colors, there is no intermediate colors. Rather, there is completely different action going on. What happens here? Both the colors they are expressing, so the blue color is due to fine alternating stripes on the feathers. These fine alternating stripes, each stripe is expressed by a particular allele. So here both the alleles are expressing to their total. So there is no mixing, no intermediate, rather each allele is acting separately and forming separate stripes. Now when the blue color that is the hybrid fowl, is crossed, F1 crossed F1, then what happens? Then we have 1/4. One of them becomes splashed white, two becomes blue, and one becomes black. As the parents we have seen splashed white crossed with black ones. So here in the F2, again we get the 1:2:1 ratio where the heterozygotes are blue. So the alleles, heterozygous alleles, both of them are expressing here and they produce blue color. I suppose all of you have found that blue color does not have any specific gene. Rather, it is an expression of both the alleles. Black color is caused due to melanin synthesis, which is caused by, or which is the expression of the homozygous recessive gene. In the heterozygous condition, in the homozygous condition means there are two doses of the allele, but in heterozygous condition, there is only one dose of the allele. So, one recessive gene produces the color. In white, there is no such allele, and so no melanin is synthesized, and they are white in color. I suppose you have understood how codominance is applying in Andalusian fowl. Another good example of codominance is the coat color of cattle. You must have heard of this word, roan coat color, roan. And this roan coat color is found in many animals, including horses, cattle, antelopes, and dogs. Roan coat color, this is an even mixture of white and pigmented hairs. White color and pigmented. These pigments can be black, can be red, or whatever color it is. And these pigmented hairs, they do not gray out or they do not fade with the aging of the animal. For example, in shorthorn cattle, a red bull, means red coat color bull, when crossed with a white coat color cow, then what happens? He yield a roan coat color calf. If you see, it may look like a blended skin color, and it may look like an intermediate color, but if you see very carefully, then you will find there are alternating separate hair colors. Some you'll find a red hair color, which is fully red, it is not blended. And another hair which is totally white, there is no blend. None of the hair will be a mixture of the two colors. So each color here expresses to its fullest. So the alleles, they are dominant. Both the alleles are dominant. So red color bull produces red color hair, white color produces white color hair. Now in the roan coat cattle, what happens? There is combinations of red hair and white hair. So roan cattle cannot breed true. I suppose you understand they cannot breed true because it is a heterozygous animal. These animals, if I cross the roan coat color animals, then again there will be segregation and you cannot get roan coat color. Yes, you can get, but the ratio again will become 50%. So best is that you, to get more roan coat color animals, the best way is to have a cross between a red bull and a white cow, and then 100% is roan coat color. This is how we can explain codominance in the cattle also. Let's talk of multiple allelism. Now, till now we have studied that for each character there are two alleles, and the two alleles means they are the traits bearing. But in multiple allelism, here we will observe that not two, but there can be more than two alleles for the same gene. That means in the same locus, there is a possibility of different types of alleles, and they will be governing the same character. This is multiple allelism. So, since in an individual we cannot find all the several alleles because there are only two sites for this. In diploid animals, there are homologous chromosomes, and here homozygous conditions can be found, or heterozygous condition can be found. That means only two locations, but there are more than two alleles. So what happens? To study multiple allelism, population studies have to be made. The best example of multiple allelism is ABO blood group, and these are controlled by the I gene. Now, what happens here? The RBC, plasma membrane, has sugar on the surface, and this sugar is controlled by the I gene. I gene has three different alleles: IA, IB, and small I. IA and IB, they produce slightly different form of the sugar, while small I doesn't produce any sugar. Since human beings are diploid organisms, so every person can possess any two of the three I gene alleles. IA and IB are completely dominant over small I. So, when IA and small I are present, naturally IA will express. Whereas, when IB and small I are present, at that time only IB will express. Reason again, small I is recessive. But when IA and IB are present together, both of them will express their own types of sugars. This is called as codominance. So red blood groups cells, they have both A and B types of sugar in the blood group. In case of blood group AB, there are both type of sugar, or it is an antigen also. Since there are three different types of alleles, there are 6 different combinations of these alleles. So in human, you can find 6 different types of genotypes. Blood group are only 4, but genotypes are 6. Let us study this table. See here. If the parent 1 be father or mother, parent 2 father or mother. So let us consider as father, mother, whatever it is, or the reverse. If one parent donates IA, another parent donates IA, then the offspring gets IA, and that is blood group A. Similarly, IA, IB becomes IA, IB, and is AB. Now, blood group a person having blood group A can have two different types of combination. Can be homozygous A, or it can be heterozygous A. When heterozygous, if one of the parent donates small I, another parent donates capital IA, then the offspring of course is A blood group, but it is a heterozygous condition. Similarly for B blood group can be homozygous, it can be heterozygous. So A and B can exist in either homozygous condition or in heterozygous condition. But AB has only one option. It is IA, IB. And blood group O, which is a recessive gene, it is expressed, that is the genotype of this expression is small I, small I. I suppose you have understood the genotypes of the offsprings. Now if I ask you a question based on this understanding, let me frame a question for you. If the father is A blood group and the mother is B blood group, so suppose there is a situation in the family like this. Father is A blood group, mother is B blood group, and the son is O blood group, and the daughter is AB blood group. That means there are 4 members in the family, and all the 4 members have different blood groups. Is it possible? This is a question for you. I hope you'll be able to answer this. You can have it. Do you know how? If the father is having this combination, mother is having this combination. That is, father is heterozygous A, mother is heterozygous B. Now, what are the possible combinations? The father is A blood group, mother is B blood group. Both of them are heterozygous. If you plot this in the Punnett square, make a checkerboard and plot it. Capital A and small I segregates. Capital B and small I segregates. After segregation, the pairing can be like this. Small I and small I pairs to form blood group O. IA and IB, IA of father, IB of mother, for example, they can pair and form blood group AB. This is how a father blood group A, mother blood group B, the daughter AB, and the son O is also possible. These are certain questions. This type of questions you may be asked to answer. Now let's come to the next topic, that is pleiotropy. Till now, we have seen that a single gene produces one effect, one gene, one trait. But now we'll study a case where one gene may affect more than one trait. This is called as pleiotropy, or these genes are called as pleiotropic genes. For example, starch synthesis in pea seed. This is controlled by one gene. This gene, starch synthesis gene, they have 2 alleles, capital B and small b. Now, starch is synthesized. Large starch grains are produced when the alleles are homozygous. That is, if the combination is capital B, then large starch grains are produced. Whereas, if it is small b, then there will be lesser efficiency in starch synthesis, and due to this, it will produce smaller starch grains. I think it is now clear. BB homozygous, small b homozygous. This produces this dominant genes producing more and large number of large size starch grains, and small b they have lesser efficiency, so they produce smaller starch grains. Now, on maturation of the seeds, what happens? Capital B homozygous, they produce larger structures or starch, and these seeds are round in shape. Whereas small b, they are less efficient in starch production. These seeds are wrinkled. Now, what about the heterozygotes? Heterozygotes, they produce round seeds because B is the dominant allele. But the starch grains, they are produced, and they are neither the large sized nor the small sized. Rather, they are intermediate sized So I think you have understood now that the same gene is now guiding two functions. One is the shape of the seed and other is the starch production. Now let us consider, if starch grain size is considered as a phenotype, then the allele show incomplete dominance. Here what we find, allele BB. This gives round seed and large size grain. The starch of size is large. Capital B, small b, these are round seed but intermediate starch grain. Small b, these are wrinkle seeded and small starch grain. So here we find that there are two characters which are being guided by the same gene. This is called as pleiotropic gene or pleiotropism. Now let's discuss lethal genes. What are lethal genes? Lethal means something causing death. Lethal weapons you must have heard of. Now these are certain genes which exert some lethal effect. So what happens? The individual dies. Now the surviving phenotype and the genotype, they get altered. So naturally the ratio will be changed. There are two types of lethal genes. One is dominant lethal gene and another is recessive lethal gene. The dominant lethal gene, for example, in mice, yellow coat color, these are dominant ones, homozygous. They die as in the embryo or at very early age, whereas the yellow, but which is heterozygous, and the black ones, they survive. So, if a cross is made between heterozygous yellow and a black one, then what happens? As we have seen in the case of moon hybrid crosses, capital Y cross with small y. This gives the F2 ratio. So it should give 3:1. That means 3 will be yellow and 1 will be black. But if you see the genotype, then what happens? 1 will be capital Y, 2 will be capital y, and 1 will be small y. Here, capital Y dies. That is the lethal effect. So the survival is only the 2 yellow, that is capital Y, and 1 black, that is small y. This is how the dominant lethal gene causes the change in the surviving Mendelian ratios. Another example, that is a recessive gene which is lethal. Recessive gene, of course, should be in homozygous condition. That means here the homozygous recessive individual will not survive. A common example is sickle cell anemia. Sickle cell anemia has two forms, normal and sickle cell. Here, the homozygous dominant gene and the heterozygous dominant gene both survive in nature. But the homozygous recessive individuals, that is, the recessive gene having individuals, they die due to severe anemia after birth or when they may die before reaching the adolescence. This is another deviation from Mendelian ratios because among the survivors, you don't find homozygous recessive individuals. Let us see this cross. This will explain it in a better way. Heterozygous HBN, HBS, normal and sickle cell. This is another individual, HBN, HBS, normal and sickle cell. So they are heterozygotes. Both of them are normal and they have heterozygous gene for the sickle cell anemia. The genes segregate, and now let us study this checkerboard. What we find here, it is the same as what we have studied in tall and dwarf. So here we find these three, 1, 2, and 3. These three are normal because they have at least one HBN. Here homozygous, heterozygous, heterozygous. And the fourth block, this one, HBS, HBS, this causes sickle cell anemia. So in this population, 3:1 is observed. But this one will not survive. So in this population, 25% population probability is there that they will die. So this is another deviation from Mendelism. In the population, the normals they survive, and the normal having a trait that is heterozygous condition they survive. So HBA or HBN will survive, and HBN HBS will survive. No chance for HBS and HBS. This is how we can explain the inheritance of sickle cell anemia. This is a recessive lethal gene. With this, we actually come to the end of today's discussion. Now, before I leave, I have some questions for you. Explain incomplete dominance with an example. Now, it is your time that you differentiate between incomplete dominance and codominance. How do you consider that APO blood group is an example of both codominance and multiple allelism? You have to explain this. Now, another question, important. There's an interesting question here. In a family, a quarrel arose on their child, who is O blood group, whereas the father is A and the mother is B. As a student of biology, how will you solve their problem and satisfy the parents? Almost the same we have already discussed. Next question is, what is multiple allelism? Explain it. So with this, we come to an end of today's discussion. I hope all of you have understood this, and I have been able to make it very clear that how there are examples where there is deviation from Mendelism. Let me again tell you, there may be deviation in Mendelism. So far, the law of dominance is concerned, but in all the cases we have studied, law of segregation holds good. Thank you very much for being with me, and I would appreciate you in my next class where I'll be discussing on linkage and crossing over. A very, very important concept, linkage and crossing over, where we'll be explaining you. And at that time, I'll explain why I have put this figure here. This photograph has some meaning. We'll discuss. Thank you very much.